Preparation of functionalized Starpeg heparin hydrogels. The first step in this protocol involves preparing glass cover slips. The cover slips must be either treated with aminosilane, used for anchoring gel substrates to the cover glass for subsequent handling, such as cell culture experiments, or they are treated with hexamethylsilane for ease of removal of the top cover glass for these anchored gel substrates, or for producing floating gel substrates. The first step involves a pre-cleaning method, in which the cover slips are placed in a Teflon shelf for all of subsequent steps, and then they are then treated in an ultrasonic water bath, first in milky water for a half hour, and then in ethanol for a half hour. After washing in milky water, the cover slips in this Teflon shelf are cleaned via an RCA cleaning method. The RCA method involves the use of milky water, hydrogen peroxide, and an aqueous ammonium solution. The Teflon shelf is placed in the RCA solution and heated up to 70 degrees Celsius. The cover slips are maintained at 70 degrees for 10 minutes, making sure that the temperature does not rise above 70 degrees. After the cleaning in the RCA solution, the cover slips in the Teflon shelf are rinsed 2 to 3 times. For the aminosilane, the cells are incubated in the Teflon shelf in an aminosilane isopropanol water solution and incubated here for 2 hours at room temperature. The Teflon shelf with the cover slips are then rinsed in isopropanol thoroughly, immediately dried with nitrogen gas, and then placed at 120 degrees Celsius in a drying oven for 1 hour. If hexamethylsilane treatment is desired for cover glasses, the cells are then dried immediately after the RCA solution. After this, the cover slips are evenly placed in a large petri dish. In a small petri dish in the center of the dish, containing 100 microliters of hexamethylsilane, is placed. The petri dish is then closed with parafilm, and the cover slips are incubated in this manner for at least 3 hours to overnight. For gel preparation, each component should be weighed into a separate microcentrifuge tube, depending on the size of the volume needed. The first step in the protocol involves adding 1/3 of the total volume of ice-cold milky water to both tubes containing heparin and PEG. For example, for 300 microliters of a total gel solution that's desired, 100 microliters of milky water should be added to each the PEG and the heparin. The PEG and heparin solutions are centrifuged briefly and incubated in an ultrasonic water bath filled with ice-cooled water for 10 minutes to fully dissolve the solid components. After this, the PEG and heparin solutions should each be kept on ice until use. After the heparin is dissolved, 1/6 of the total volume of ice-cold milky water should be added quickly to the EDC and NHS components. For example, for a 300 microliter total gel solution, 50 microliters of milky water should be added to each the EDC and NHS separately. Keeping the solutions on ice, pipette several times to fully dissolve each component, the EDC and the NHS. The EDC solution should be added immediately to the heparin and mixed well by vortexing for 5 seconds. Then, the NHS solution should be quickly added to the heparin EDC mixture and again mixed well by vortexing for 5 seconds. The heparin is activated in this manner by incubating on ice for exactly 15 minutes. After 15 minutes incubation on ice, the PEG solution should then be added to the activated heparin solution. This should be mixed immediately by vortexing for 15 seconds. Immediately, one should pipette the desired gel volume onto each cover slip and carefully place the top cover slip, functionalized side down, on top of the solution. For anchored gel substrates to the glass cover slips for subsequent in vitro experiments, it is easier to pipette the liquid gel solution onto the hydrophobic or hexamethylsilane treated cover slips, and then cover the gel with the aminosilanized cover slip, gel side down. For the non-anchored or floating gels, the gel solutions should be sandwiched between 2 hexamethylsilanized treated cover slips. The gel should be allowed to form at room temperature overnight in a parafilm sealed humidified chamber. The humidified chamber is made by wetting a small piece of chem wipe or tissue with milky water and placing this in the petri dish with the cover slips and gel solutions. After polymerization, the hexamethylsilanized cover slips should be carefully removed from the solid gel. The gel should be placed in a well plate or other dish suitable for the size and hydrated in sterile PBS for at least 24 hours before use. The PBS should be exchanged several times over this time period to efficiently remove any of the unreacted components. To further sterilize the gels, they may be placed under a UV lamp for 30 minutes and or stored in a 2 millimolar sodium azide in PBS solution overnight. For functionalizing the Starpeg heparin gels with RGD or other amine-terminated peptide sequences, the gels should first be rinsed 3 times in an ice-cold 1/15 molar phosphate buffer and incubated at least 10 minutes. The activating solution should then be prepared by dissolving the EDC and NHS separately in an ice-cold 1/15 molar phosphate buffer. Each component should then be sterile filtered and then mixed together in the appropriate ratio. The gel should be incubated in this ice-cold activating solution at 4 degrees Celsius for 45 minutes. Gels should be rinsed briefly 3 times with 100 millimolar borate buffer. Then, the RGD solution should be immediately added to the activated gels. They should be incubated for 2 hours at room temperature in this solution. The gel should be rinsed 3 times with sterile PBS. At this point, they could either be functionalized with cytokines or growth factors, or they could be used as they are. Good luck!